Hello students, today we are going to discuss about enzymes. We are going to take into consideration the biomolecules, that is an enzyme which is made up of proteins. Now let us see what actually an enzyme is all about. When I talk about enzymes, I am talking about biological catalysts, which means a catalyst which is being synthesized inside a living cell. When I say biological, that means it would be synthesized by a living cell. And when I say catalyst, from your knowledge of chemistry, you must be knowing a catalyst is a substance which hastens up the reaction. Means if there is a chemical reaction or biochemical reaction, it would speed up the rate of reaction. So we have two types of catalysts. Few are organic catalysts and mostly there are inorganic catalysts. Now you have to identify the difference between the two, that these catalysts, you are dealing with these in your chemistry processes, that is whenever we are talking about chemical processes, and these catalysts often require high temperature. Try to recall the reactions you do in your chemistry projects and chemistry. It basically suggests that there would be a catalyst which would be present, and that catalyst would require a lot of high temperature or pressure. But when I am talking about organic catalysts, it would be an enzyme specifically. That enzyme would not need lot of high temperature. So when I talk about biological catalysts, I am referring to the enzymes, and if I talk about those enzymes, they are mostly made up of protein. Mostly would not be a correct term, they are basically made up of protein only. So there is a sentence as well which identifies what basically enzymes are made up of. All the enzymes are mostly proteins, but all the proteins are not enzymes. So if I say that how the enzymes are formed inside the cell, get back to your knowledge of protein synthesis, that is done by DNA. So basically, the DNA of a cell contains the blueprint for the production of all the enzymes which would be required for the activity of cell. And why these enzymes would be required for activity of cell? For the various metabolic processes. You might remember that we talk about kinase, pyruvate kinase, catalase, and many enzymes are those which end with a term ase. Alright, supposedly this is something, and if I say lactase, that means this is an enzyme which would be responsible for breakdown of lactose. So this ase term is used for enzymes mostly, and these enzymes are made up of proteins. Okay, it is synthesized inside the cell. The blueprint lies with the DNA. It is proteinaceous component. Apart from proteinaceous components, there are certain things known as ribozymes. Please cram this term. What is a ribozyme? Not just proteins, at times certain nucleic acids like RNA can act as an enzyme. So basically, when RNA acts as an enzyme, means RNA is acting as a catalyst, it is showing a catalytic activity, it would be known as ribozyme. This is one term you need to remember. Coming back to the basic properties of a particular enzyme, we did talk about that it is made up of proteins. Those proteins would be synthesized in the cell. Now, what is the function of these biological catalysts? Why do we need these biological catalysts inside a cell? We need these catalysts inside a cell so that the metabolic reactions which would otherwise take at least 3 years or 4 years for completion, they get completed within seconds. Alright, try to understand what I am telling you, the extent of the speed that they provide to the reaction. Supposedly a reaction is to be completed within an hour, an enzyme can speed it up to 10 million times. This would be something that you take round of entire earth in blink of an eye. That much would be the speed that an enzyme provides within the cell to a reaction. Alright. So when we talk about the basic composition, we say that it is proteinaceous, or it could be a nucleic acid, as in the case of ribozyme, that would be acting as a biological catalyst. Apart from the proteinaceous part, the enzyme can have prosthetic group along with it. Alright, so the question arises, what is a prosthetic group basically? Supposedly this is an enzyme, this much part is protein. Now, one thing more you need to understand, that enzyme would also have a secondary or a tertiary structure like a protein, that on a backbone there would be a protein which would be coiled, and there would be certain holes and crevices which would be created in the structure that we would come to later on. But for now you need to understand that the proteinaceous part, which could be secondary or tertiary, may have some non-proteinaceous part. Now this non-proteinaceous part happens to be prosthetic group. Okay. Now what is the need of this prosthetic group? These prosthetic groups are also responsible for providing specificity and other general properties to an enzyme, due to which it is able to catalyze the reaction. Okay. Now these prosthetic groups are further of depending on the type, that what has come into attachment with the proteinaceous part of the enzyme. Prosthetic group could be a metal compound, as in the case of iron porphyrin compounds that are present in cytochromes. Okay. Or it could be, these are metal compounds, or it could be organic compounds, as in the case of NAD. You remember what is NAD and FAD? This is precursor to niacin, and this is precursor to vitamin B12. Okay, these are the prosthetic groups which go and join with the enzyme. Alright, it could be a metal compound, it could be an organic compound, it could be a metal ion as it is, as in the case of iron ions which combine with the catalase for its activity. Okay, catalase is an enzyme. As the name suggests, it must be enabling some sort of catalysis inside the cell for a reaction. So that is a prosthetic group, non-proteinaceous part, which would come and combine with the protein. Okay, and that protein would be acting as an enzyme. These together would act as an enzyme and enable the catalysis. Now these metal compounds would be known as prosthetic groups, and these organic things which come and combine with the enzyme, they are also known as coenzymes. Okay, so we come across the term cofactors and coenzymes. What are cofactors and coenzymes? Those organic compounds which would come and join with the proteinaceous part of the enzyme, that would be known as a cofactor or the coenzyme. Now let's come to the mechanism of action. How does an enzyme enable the catalysis? To an extent which I said that at least 10 million times, like the speed of the reaction could be enhanced up to 36 million times. So how is an enzyme, or if we call it a protein, able to hasten up the reaction to such an extent? So the answer lies behind this particular thing, that is the activation energy. Now from your knowledge of chemical kinetics, try to recall that behind every reaction, if it is biological or it is chemical reaction, there is an activation energy. The entire reaction study deals with a term known as free energy. To make it simpler, not bringing more of physics and chemistry into your biological knowledge, consider it as potential energy. Okay. Now, according to thermodynamics, every compound has an inherent capacity to lower down its potential energy or to lower down its free energy. That means a thing is stable if it is having a lower free energy. Now when I talk about activation energy, whenever the reaction has to take place, there would be reactants which need to reach to such an energy from where the reaction has to take place, because ultimately reaction is breaking and forming of chemical bonds. So the reactants will undergo a transformation where they would reach the activation energy, which could be provided externally, or they would develop that internally so that the bond breakage is easier. So these reactants have to reach the activation energy, and here where the enzymes are going to act. This is the place the enzymes would come into action. They are going to reduce this activation energy to a very low amount, so that reaching a lower potential energy or reaching a lower free energy is easier for the compound. Okay. This is just an overview I'm trying to give you. Supposedly there is a reactant, it is having high free energy, okay, it is at a high free energy state. Its tendency would be to come to a free energy where the free energy condition would be lower, or the potential energy would be lowered. For that it needs to undergo a reaction, a chemical reaction where there would be formation or breakdown of the bonds. For that, first of all, an activation energy, that is a zenith, it must reach, the activation energy must be achieved, and then the chemical reaction would basically take place. That would involve the breaking and making of bonds, and that would lead to a condition where the substrate which would be formed. Now here another term, product, sorry, it's not substrate, it's product. A product would be formed as a result of the reaction, and it will have a lower free energy. Now in terms of enzyme, we use the term substrate. We come to know a new term, that is active site. As I said, the enzymes are proteins and they have a three-dimensional structure. So when we discuss their three-dimensional structure, it is a protein that has taken curves around itself, and it has coiled to an extent that it forms a tertiary structure. So there are certain holes and places which are formed in that 3D structure. Supposedly this is an enzyme. This is an enzyme, and this site over where the hole and a crevice could be created is known as active site. Okay? This is the active site. Now whichever product is to be formed, that would be formed from a reactant that is used as, that is referred to as, not used to, that is referred to as substrate. Now there are many active sites in the enzyme. In an enzyme there are many active sites. Now it depends on the affinity of the active site towards the substrate, which decides how fast the reaction would be, or how well the enzyme would be able to catalyze the reaction. Now the substrate comes and binds with the active site, and it is very much responsible for the specificity of the enzyme, that a substrate with specific shape, as you can see this shape, the substrate, if it is of this shape, it will not be able to fit into it. It has to be in correspondence with the active site. Okay, it comes and joins with the active site. Now what happens over here? The dynamics which are happening behind this particular formation, we can represent it graphically. For that, over here I am going to consider free energy, okay, or you can take potential energy, and this is time. Okay, we are considering this particular reaction, how it takes place. Now see one thing, supposedly the substrate was at this level of the free energy. Now as we discussed earlier, the activation energy is usually higher. So this activation energy is till here. Now the substrate has to increase its energy till here, so that it comes at a stage where the free energy is as low as this point, and over here we get the products. Okay, we have a substrate, it is having a free energy higher than that of product. The reaction proceeds only at one condition, that the product must have a free energy quite lower than that of a substrate. Now this substrate has to reach this peak of activation energy, and then the reaction would proceed and this particular product would be formed. Now this activation energy state is known as, where the compound has to reach this activation energy, this is known as transition state. Okay. Now what happens when the enzyme comes into being, when the substrate combines? Now supposedly you are providing this substrate energy from outside, some sort of temperature you are increasing, then you can reach this activation energy, as I am telling you. But when the enzyme comes into scene, this activation energy is lowered down to a great extent. Okay, and then the reaction further proceeds in the same way. Only thing that is happening is that this activation energy, which was quite higher, the transition state which was achieved at a quite higher energy, that would be achieved at quite lower energy now. Now this process is done by the enzyme which acts as a catalyst. Now the substrate would combine at the active site, then we will have ES complex which would be formed. Okay, when the combination takes place, then we have ES complex. This ES complex would bring down the activation energy. The transition state has come at a very low free energy. The ES complex, the substrate would undergo the changes, and as soon as the reaction is completed, this substrate would form a particular product. Supposedly this is the product, and the enzyme would be ready for another reaction catalysis. Is it clear now that how this substrate it combined with the enzyme, the substrate formed the enzyme substrate complex, that enzyme substrate complex later gave rise to the product. Seeing it in this graphical presentation, this is the free energy of a reaction which is not taking place in the presence of enzyme. A lot of activation energy is required in this case. This is the activation energy. When the enzyme comes into being, ES complex, this reduces the activation energy to this level, and the reaction proceeds and the products are formed at a much speeded up rate. The entire reaction takes place at a very fast rate. The rate of the reaction changes, and this is what the enzyme is supposed to do, that is to enhance the rate of reaction to a great degree, so that